Hello everyone. Today we are going to be talking about glycolysis. So, firstly, what exactly is glycolysis? Um, the main point of breaking glycolysis, or the main point of glycolysis is taking a single monosaccharide or a single sugar, um, which is glucose, and breaking it down into two pyruvates. Um, as this reaction occurs many times, the main point of glycolysis is to create more ATP and create electron carriers along the way. There's a huge process of this, from glycolysis to pyruvate oxidation to TSA cycle to oxidative phosphorylation, which is the electron transport chain where all of the electron carriers are transported to and facilitated in order to make more ATP, and the process is really long. This is the first part. Depending on whether it's an anaerobic whether it's anaerobic respiration or a aerobic respiration, ATP is still going to be made. So firstly, let's talk about what glycolysis really is. So firstly, to get in, by the way, this whole process occurs within the cytoplasm. And let's start. Okay, so basically a glucose monosaccharide will get within the cell, not through simple diffusion, but a specific type called facilitated diffusion. This is with a protein channel or protein carrier, if you guys would like to say. So basically, the glucose would get in. You know why? You know when we were talking about macromolecules, each the phospholipid bilayer heads on top are hydrophilic. On top of that, they have a negative charge because they are connected with the phosphate groups. What does that equal to? Large molecules such as glucose repel those charges on those phosphate and glycerol heads of those phospholipid bilayers. This leads to the fact that glucose cannot go from a high to a low concentration. They need a protein channel in order to facilitate that process. So, okay, glucose gets in through facilitated diffusion. Next, what happens? Glucose is a 6-carbon sugar. It's a hexose monosaccharide. What happens after? So, firstly, there's a lot of terms we should know before we even start. Firstly, there's things called, there's the process of redox reactions all throughout this huge complex chain. Redox reactions include oxidation and reduction. Oxidation is the process of removing an electron from a compound or molecule, and reduction is including or getting a form of electron attached to another molecule or substance. So reduction is basically the opposite of oxidation. Reduction just likes to add electrons to substances and molecules. So okay, and another one is ATP, ADP, and etc. So basically, when a phosphate group is removed from ATP, when a phosphate group is removed, it's called dephosphorylation. And when a phosphate group is added, it's called phosphorylation. When it goes from ADP to ATP, it's called phosphorylation. So let's start. Firstly, the 6-carbon molecule. The first step right off the bat is the fact that the 6-carbon molecule, when it starts the process of glycolysis, it gets phosphorylated automatically. Our body sends in 1 ATP, and that ATP gets dephosphorylated. And that means it loses a phosphate group, and guess what happens? That phosphate group attaches right over here. So basically, that's the first step that even happens. So within this process, there's many processes that are reversible and non-reversible. We'll talk about them. So the first thing that happens is the fact that glucose contains a phosphate group on its first on its first carbon on its first carbon on its first carbon atom. So there's two types of enzymes that facilitate this process: hexokinase and glucokinase. Glucokinase is found within liver, and hexokinase is found within muscles. So as they facilitate this process, by the way, if you don't know how enzymes and other basic processes work, please refer to the glossary, as many processes within glycolysis are very, very complex. Anyways, so basically, enzymes catalyze this reaction, and the first enzyme within this process is called phosphohexose isomerase. This process basically means that the arrangement of the atoms within this molecule, this 6-carbon molecule, is turned into fructose 6p. From glucose 6p, it turns into fructose 6p. So glucose 6p is a form of aldehyde, fructose 6p is a form of ketone. And basically, this enzyme right here catalyzed that and basically isomerized glucose. So the next step that happens is from when fructose 6p is turned into fructose 1,6-bisphosphate. What happens here? What happens during this process? Another same process happens once again. ATP is dephosphorylated once again into ADP. That phosphate is attached to the carbon 6 on fructose 1,6-bisphosphate. Why is it called bisphosphate, by the way? Until now, we've heard biphosphate. Why is it called bisphosphate? This is due to the fact that there are carbon links between the chemical arrangements. So the enzyme that facilitates this process and catalyzes the whole reaction is called phosphofructokinase. So after fructose 1,6-bisphosphate is in place, this is the time where we have to split this. All right? To split this, an enzyme named aldolase comes in and splits fructose 1,6-bisphosphate and separates the phosphate groups to separate molecules. The first one is DHAP, or dihydroxy acetone phosphate, and the other one is glyceraldehyde 3-phosphate. So firstly, we need to realize that, look, this side, you can almost think of it as not really reliable, because firstly, DHAP, we're just going to refer to that as DHAP. We're going to refer to this as GA3P. So DHAP and GA3P are convergible or interchangeable, you can say, or interchangeable with one another, vice versa, as we need to get DHAP all the way down to this side in order to make pyruvate. So what enzyme allows that? The enzyme called triose P isomerase allows this process. So DHAP interconnects and interchanges with GA3P, causing each reaction to have two of everything. So firstly, let's look at what happens. As GA3P wants to turn into 1,3-bisphosphoglycerate, a big process occurs here, all right? This process is caused by an enzyme named glyceraldehyde 3 dehydrogenase. Basically, what it does, firstly, it does two things. The first thing it does is it allows electron carriers, these two NAD pluses, to be reduced. Remember what we said, guys. Again, reduce means they are getting attached with electrons. So these two NAD pluses are getting reduced with hydrogens and they make two NADHs. These two NADHs are already electron carriers that are ready to go to the electron transport chain. However, they cannot yet because a process still has to occur. So the second job of glyceraldehyde 3D hydrogenases, you see this inorganic phosphate that's just roaming around? It likes to add that within the process, and that phosphate got linked between without dephosphorylation or phosphorylation of any ATP whatsoever. And the new formation of this is 1,3-bisphosphoglycerate that has, as you can see, 1 is for the phosphate group on the 1 carbon, 3 is for the 3 carbon, is the phosphate group connected to the third carbon. So now what happens is 2 ADP gets phosphorylated to 2 ATP. What does that mean? That means when we phosphorylate it, we lost a phosphate. That phosphate is gone now. So now that phosphate is gone, the enzyme that conducted this and facilitated this was called phosphoglycerate kinase, or you can also call it phosphoglycerate 1,3-BPG. It's a really long enzyme, but however, it does not make a big difference knowing the name. It's more important learning how the process works itself. So basically, and just to rephrase it again, if you still don't know how to spell it, phosphoglycerate kinase, basically. That's the process. That's the enzyme that facilitates from 1,3 bisphosphoglycerate to 3 bisphosphoglycerate. So what's the process that just happened right now? We lost a phosphate group. Now it's just 3 bisphosphoglycerate. The one is gone because the phosphate group is gone. So now another enzyme happens. Another enzyme facilitates this process too. It's called phosphoglycerate mutase. Guess what this does? Guess what this does? I just realized something. My apologies. I forgot to address it earlier. When we were talking about the reduction of NAD+, we didn't talk about what it was reduced with. So my apologies for that. The enzyme here, glyceraldehyde 3D hydrogenase, it basically helps the two NAD pluses take off hydrides off of the glyceraldehyde 3-phosphate, or GA3P. This enzyme allows NAD pluses to take hydrides off GA3P. Hydrides basically have 2 electrons and they are positively charged within the middle. So as they are connected, that's how they get reduced, and that's how they became 2 NADHs. Sorry for that. Sorry for not clarifying that earlier. Now back where we were. This 3 bisphosphoglycerate gets converted to a phosphoglycerate. It gets converted to a phosphoglycerate because of one main thing. This phosphoglycerate mutase, you know what it allows for this process to, you know how it happens? It's because there is a organic compound called enol that has the chemical structure of a carbon bond, one carbon bond, and a double carbon bond with another carbon, and it has another bond with a oxygen. That oxygen has a phosphate group. It's amusing, isn't it? It's really crazy knowing that an enzyme can just change the formation like that straight up. Anyways, basically the phosphate group is attached to the oxygen here. That's why it's in the middle. That's why the formation from 3 bisphosphoglycerate goes to 2 phosphoglycerate, because the second one, because it's in the second carbon. It's not within the third carbon anymore. So now after this process, what comes next? The enzyme enolase makes the final intermediate process possible. What it basically does is a very simple process. So my hands are really sweaty. Apologies. Basically, within the last process over here, it's called PEP. It's called phosphoenol pyruvate. This is enolase basically makes preparations for the final for the final process for PEP to be the final intermediate for pyruvate. So enolase allows that to happen, and basically phosphoenolpyruvate or PEP gets converted to pyruvate, or PEP gets converted to pyruvate by an enzyme called pyruvate kinase. And guess what? Guess what happens at the end? Due to a phosphorylation reaction, the phosphate group from the second carbon is gone because ADP phosphorylates this reaction by the enzyme pyruvate kinase and takes that phosphate away. That's pretty crazy. So now that we talked about how everything led to the formation of pyruvate, let's look at how many pyruvates there are. You know, when we were talking about how many different types of when we were talking about this GA3P here, this GA3P right here and this DHAP right here, you know how they were convertible or interchangeable? Basically, this main DHAP, it can't really continue the process further here, so it has to continue the process here, and everything turns into 2. That's why you can see a 2 in front of almost every single process that we were talking about now. That's why there's 2 pyruvates instead of just 1. So the final process, I'll say it once again. PEP is catalyzed by the enzyme pyruvate kinase, and it allows the phosphorylation of ADP to ADP to ATP possible, and the phosphate group is officially gone. Now there's 2 pyruvates, and that's basically the process. We're going to be talking about anaerobic respiration and aerobic respiration in another video. However, hopefully you got the general concept of how glycolysis goes all the way from glycolysis to pyruvate. Thank you.